Hello friends, now today we are discussing about post-translational cleavage of viral proteins. In our last lecture, we discussed about glycosylation of enveloped proteins. The glycosylation is also triggered by few polypeptide chains that is about 15 to 30 amino acids long. So in this case, the cleavage of protein or polypeptide is very crucial for different purposes, like first is the glycosylation or the infectivity of virion. So in case of picornaviruses, flaviviruses, togaviruses and caliciviruses, they have basically polycistronic mRNA, and during the translation, when the protein or polypeptide chain that is attached to the ribosome, the cleavage is initiated and the few polypeptide chain that trigger for the glycosylation. So these are the proteases that are required for the cleavage of this protein. The proteases are trypsin-like proteases, pepsin-like proteases or pepin-like proteases. It is also called serine or cysteine proteases, aspartyl proteases and thiol proteases. So basically these proteases are required for cleavage of protein. Sometime when the transcription takes place, the polycistronic mRNA chain is to be prepared, and this polycistronic mRNA produce a single protein. So now this single protein is to be required to cleave in monocistronic proteins. So this is one example. The polycistronic mRNA produces polycistronic protein, and then in case of picornavirus, flaviviruses, togaviruses and caliciviruses, this polycistronic protein is to be cleaved. Now in case of monocistronic mRNA, where already the mRNA translate monocistronic protein, or we are calling it as a small polypeptide chains, so it doesn't require any cleavage. But again the cleavage is essential because for the viral infectivity. So in this case, the cleavage of the protein is to be takes place in Golgi bodies or in any transfer organelle that transfer the protein to the membrane. So here, for example, in case of the hemagglutinin and fusion proteins, they must be cleaved for producing viral infectivity. So there is a big difference in polycistronic mRNA and in case of monocistronic mRNA, that in case of polycistronic mRNA the cleavage takes place in the ribosome, but in case of the monocistronic mRNA the cleavage of protein takes place either in the Golgi body or any transferring organelle that transfer the protein to the cell membrane. So hope you will enjoy my lectures, and if you like my videos, please subscribe my YouTube channel. Thanks.